This is a video introduction to enzymes. Enzymes are catalysts, so let's start out by reviewing what we already learned about catalysts. Let's start out by drawing an exergonic reaction. The activation energy of a reaction is represented by the difference between the reactants and the transition state, as it's shown on this figure. A catalyst lowers the activation energy by lowering the energy of the transition state. So it has a smaller activation energy, and it makes the reaction happen faster. The activation energy represents the energy that it takes for reactants to collide in the correct orientation with enough energy to break and reform bonds. So this energy that we call activation energy is the energy associated with bringing molecules together in the correct orientation and in breaking bonds. Catalysts lower the activation energy by reducing the energy of these three things. They can help bring molecules together, they can help put them in the correct orientation, and they can help break the bonds. Enzymes are catalysts, and we are going to consider hexokinase as our example. Hexokinase is the enzyme that catalyzes the reaction of glucose and ATP to form glucose 6-phosphate and ADP. This is a critical part of metabolism. I've written the reaction here in two different ways. The lower box is the cartoon of this reaction that you'll find in your textbook, and the upper box is the actual reaction with chemical structures. I've tried to highlight the different parts in the same way that they're highlighted in the cartoon in your textbook. Glucose and adenosine triphosphate, or ATP, react to form glucose 6-phosphate and adenosine diphosphate, or ADP. Notice that the thing that happens in this reaction is that a phosphate group is transferred from ATP onto glucose. The T in ATP, or the tri in triphosphate, stands for the fact that this molecule has 3 phosphate groups. When one of the phosphates is transferred to glucose, this is now called adenosine diphosphate, or ADP, where the D or di stands for 2 phosphates. Hexokinase catalyzes this reaction by bonding to glucose and ATP and helping to convert them into products. Now we're going to see a video of how this happens. This is the same video that you can find on your Sapling Learning website. Hexokinase will bring ATP and glucose together by binding them in an active site. The active site allows the reaction to happen. It brings the molecules close together in the correct orientation for the transfer to happen. It also helps to break the bonds. Once the products form, the enzyme releases the products, ADP and glucose 6-phosphate. This reaction, under normal conditions, would take 10,000 years. However, in the presence of an enzyme like hexokinase, it happens thousands of times per second. This is a huge increase in reaction rate, from 10,000 years to 10,000 times per second. That's pretty amazing. Biological molecules like enzymes are specially designed to carry out specific biological reactions very, very quickly in your cells. Let's take a closer look. The steps of this reaction are shown here. First, the enzymes and substrates are present together, and then they form an enzyme-substrate complex. Once the enzyme-substrate complex forms, the enzyme can convert the reactants into products and release them into enzyme plus products. Although this enzyme adds an extra step to the reaction, it actually makes the reaction faster by lowering the activation energy. Here's the uncatalyzed reaction. The catalyzed reaction involves an extra step, the formation of the enzyme-substrate complex. However, the activation energy barrier for each of the steps is lower than the overall uncatalyzed reaction. In this case, the activation energy would be the difference between the reactants and the highest step. This is the activation energy of the enzyme-catalyzed reaction. Even though there's an extra step, it's still a faster reaction overall than the uncatalyzed reaction. Enzymes are amazing because they can make reactions happen much more quickly than they would in ordinary circumstances. They are even more interesting because they only react with specific reactants. This reaction will be catalyzed with glucose and ATP. but the enzyme will not accept any similar sugar molecules. So an epimer of glucose would not work in this reaction. In fact, this enzyme is specific for D-glucose and will not accept L-glucose into the active site. The active site is the part on the enzyme that facilitates the reaction, and it's the part on the enzyme where the reactants bind. The active site of hexokinase has specific amino acid side chains that are perfectly positioned to hydrogen bond to glucose. These amino acids are lysine, threonine, glutamine, asparagine, and aspartate. Because of their positions in space, D-glucose fits perfectly and hydrogen bonds perfectly in this active site. This is why we can say that hexokinase is specific for D-glucose. Any other stereoisomer of glucose, even if it's very similar, it will not have all of the correct orientations of the atoms to fit perfectly in the active site, and this will inhibit the reaction. Here's a drawing of the actual structures of the side chains, hydrogen bonding to glucose. It's hard to see the three-dimensional shape here, so you'll have to use a little bit of your imagination. But it's important to understand that the shape of the active site is specific for the substrate. And when we're talking about enzyme-catalyzed reactions, the substrate is simply the reactant. Here's another picture of the active site from your textbook. This is a cartoon version. This also shows that there is a magnesium in the active site. A magnesium ion is bound to the active site, and the magnesium ion interacts with the phosphate ions on ATP through ionic interactions. And it holds the phosphates in just the right orientation with respect to the glucose so that the reaction can happen. This magnesium also polarizes the bonds and helps them to break. Once the products are formed, they don't fit well in the active site anymore, and therefore they are released, and the reaction can happen all over again once a new molecule of ATP and glucose bind in the active site. There are two binding models for enzymes that you should know about: the lock and key model and the induced fit model. In the lock and key model, the enzyme is prearranged in the perfect orientation for the substrate to bind. Notice in this figure that the enzyme has a complementary binding site to the substrate, and the shape of either one of them does not change when the enzyme-substrate complex forms. The second model is the induced fit model. Notice that the enzyme has an active site which does not perfectly fit the substrate. When the substrate binds to the enzyme changes its conformation to fit the substrate perfectly. The hexokinase catalyzed reaction that we looked at earlier fits the second model, the induced fit model. Let's look at the video one more time and try to identify why hexokinase fits the induced fit model. Here's hexokinase. Glucose and ATP will bind in the active site. Notice what happens when they go in there. The enzyme changes shape to fit the substrates. Once the enzyme fits the substrates, the reaction can happen, and then the products no longer fit in the active site, so they are released. This is hexokinase, and it fits the induced fit model of enzyme-catalyzed reactions. If you'd like to view this video again, it's available on your sapling activity. Each enzyme will have an optimal pH, an optimal temperature, and a maximum activity. Let's talk about what this means. On your activity, there are 3 different charts that I'd like you to fill in. First, let's talk about optimal pH. Each enzyme will have an optimal pH at which it is most active. Activity is the conversion of substrate into product. So if I were going to complete this chart, I would make sure that my highest activity is at this optimal pH. As you already know, changes in pH can denature proteins. And so there will be ranges that are very low and very high where the protein would be denatured. In this window, the enzyme would be active when it was folded properly, and the optimal pH would be its most active pH. So this might be what an activity versus pH chart would look like. Enzymes also have optimal operating temperature. Body temperature is usually the optimal temperature for enzymes because they have evolved to operate at our body temperatures. So in this chart of activity versus temperature, the maximum activity should be at body temperature. As you've learned, As you approach 50 degrees Celsius, you denature proteins. And so above 50 degrees Celsius, the protein would be denatured and it would not be active at all. Temperature represents energy. At very cold temperatures, reactions slow down. And so there would be a range at the low temperature end where it would be too cold for the reaction to happen. Once again, we have a window of activity where body temperature would be the maximum. This is what a chart of activity versus temperature might look like. At a certain temperature, there would be enough activity for the reaction to begin happening. You would increase activity until you reach body temperature. And then as you get closer and closer to 50 degrees, you may start getting too hot until the point where you denature the protein. And they don't have any activity under denatured conditions. Each enzyme also has a maximum efficiency or a maximum activity. It can only work so fast. Your textbook uses an analogy of workers and bicycle parts. No matter how many parts you have, if you only have 3 workers, they can only make so many bikes per day. Enzymes work the same way. If you increase substrate concentration, you can increase activity until a certain point. The concentration that's associated with maximum activity is called the steady state concentration. After this concentration, increasing the substrate concentration does not increase the activity because you've already reached maximum activity. Please take a moment to think about these things and make sure they make sense to you. Answer the questions on this page of the activity and check the key to make sure you're correct. Now I'd like to talk about enzyme inhibition. There are molecules that will inhibit enzymes in 2 different ways. The first way is through competition. A competitive inhibitor will be similar in shape or in functional groups as the substrate. This is a cartoon of an enzyme that has a square substrate binding site. This is the substrate and this is the inhibitor. Notice in my cartoon that they have the same shape. As far as chemistry goes, this means usually they have the same functional groups. The substrate and the inhibitor would fit into the same site, and so they are in competition. This is a competitive inhibitor. The inhibitor is similar in shape or in functional group to the substrate, and therefore the inhibitor can bind competitively to the active site where the substrate usually binds. The other type of inhibitor is a non-competitive inhibitor. Notice in this cartoon of a non-competitive inhibitor, the inhibitor has a different shape than the substrate, or perhaps it would have completely different functional groups. The substrate and the inhibitor in this case bind to completely different sites because the inhibitor would not fit in the same site as the substrate. So the substrate fits here and gives you the enzyme substrate complex, and the inhibitor binds to a different part of the enzyme, which changes the enzyme so that the substrate cannot bind. Notice as this inhibitor binds to the enzyme, the shape of the substrate binding site changes and therefore the substrate cannot bind. In a non-competitive inhibitor model, the inhibitor is a different shape and binds to a different site than the substrate. Now let's look at an example of 2 molecules. Succinate is the actual substrate for an enzyme, and malonate is an inhibitor. Look at the functional groups and look at the similarities between these 2 molecules and determine whether malonate might be a competitive or non-competitive inhibitor. Please take a moment to answer the questions on this part of the activity and check the key to make sure you're correct. Thanks for watching this video introduction to enzymes. If you have any questions, feel free to post them in the comments box. And if you found this video helpful, please let me know by giving me a vote.